Hello everyone, welcome to my channel. Today we are going to discuss about color vision, color blindness, and answer the major question how we see color. So in this section, sensation and perception, color vision and color blindness, it is very important topic. Let's discuss about it. Color vision. It is our ability to perceive differences between light of different wavelengths or frequencies, which is totally independent of light intensity. So, if we talk about the light, it can be red, blue, green, violet, indigo, and these light have got its different wavelengths. So color vision, it is our ability to perceive that differences between light of different wavelengths, and it is completely independent of light intensity, and it's complex process also. Why? Because it involves the eye and the brain. The visible light spectrum, as you can see in a figure, it is a section of electromagnetic radiation that is visible to human eye. So here, if you see the figure, the difference in the wavelength of each color, like we can see violet, it is between 380 to 435 nanometers, and if we see red color, we can see the differences in the wavelength, that is 625 to 740 nanometer. So each color has got different wavelengths as defined in previous slide. Let's answer the major question how we see color. So how we see color or color perception starts in the retina. If you see the anatomy of human eye, this is the structure of retina. So when we see at the retina, color perception starts in the retina at the back of eye, which is at the back of our eye, with specialized light sensitive cells called photoreceptors, and these two are the major part of the photoreceptor cell, that is cone and rod. Cones, it is centrally located, whereas rod, it is peripherally located. So when we see this small section of retina, we can see the structure of cones as it is enlarged in this figure. So cones, if you see, it's a structure and differences in the rod. These are the photoreceptor nerve cells. Cones. These cells are responsible for color vision. Number one, it is responsible for color vision, and it works best in brighter light. Humans typically have three types of cones, which is why we have trichromatic vision is sensitive to different wavelengths. As you can see in the structure, there are three types of wavelength, that is short wavelength, medium wavelength, and long wavelength. In short wavelength, you can see most sensitive to blue light, that is as cone is sensitive to blue light, and medium wavelength M cone, it is most sensitive to green light, and long wavelength L cones, they are most sensitive to the red light. Here you can see in the figure this blue lights, this is short wavelength, and this is medium wavelength, you can see the green, and the third one is red one, which is alcones. Rods, these are much more numerous and sensitive to low light levels, but cones are sensitive to brighter light levels. Rods can only detect light intensity, which are black, white, and gray, and they play little to no role in color vision. Talking about the color perception, the color we see is determined by the ratio of stimulation among three types of cone as described in for the before slide. For instance, yellow light stimulates L cones, M cones. It is green strongly, but S cones blue only weakly. So, theories of color vision. There are two complementary theories, that is trichromatic and opponent process theory. In trichromatic theory, it is accurate for the cones in the retina, which we discussed in previous slide, while the opponent process theory is accurate for how cone signals are combined and processed further up in the visual system, that is in the brain. Let's talk about the theory briefly. Number one, trichromatic theory, founded by Helmholtz, and it is proposed stage of vision, that is usually we see color through the photoreceptor, that is receptor level cones. Color is based on the proportionate stimulation of three cone type, that is red, which is Al cone, green M cone, and blue, it is the S cone. This explains how we initially detect different wavelengths of light. But opponent process theory, it is processed in antagonistic pairs by the nervous system. This explains we don't see colors like radius green or yellowish blue. The three opponent channels are red versus green, blue versus yellow, and black versus white, to which we also say the luminance. We will discuss about this theory in upcoming videos in detailed manner, but here we have described it briefly. Next is color blindness or color vision deficiency. So color vision deficiency, it is the inability to distinguish between certain sets of color. It is often an inherited genetic condition. Number one, the major one is red-green color vision deficiency. It is very common, and anomaly or deuteranopia, it is the difficulty in processing between sets of red and green due to the issues with M cones, and deuter anomaly is the most common form because it is the mild shift in sensitivity of M cones, but deuteranopia it is severe, that is lack of M cones. You can see even in the figure here in deuter anomaly, you can see that M cones, it is mild shift in the sensitivity, but here deuteranopia, you can see that it is the lack of M cones. Next is protanomaly and protanopia. So it is difficulty distinguishing between the red and green due to issues with alcones. Protanomaly is reduced sensitivity, and protanopia is total lack of alcones. Here you can see in the figure like protanomaly. Protanomaly, you can see here the red color is, it is mild shift over here, that means alcones. It is reduced to the sensitivity, but here protanopia, you can see total cross in the red color, that is lacking of the alcones. Next one is blue yellow color vision deficiency, which is rarer. As you can see in the treat anomaly and treat anopia, it is difficulty distinguishing between the blue and green and between yellow and red due to the issues with the S cones, that is blue color. You can see in the figure treat anomaly. Here you can see the shift in the blue color, that is S cone, and here is the lacking of the S cone in treat anopia. Third one is complete color vision deficiency, which is very rare. It is monochromancy, which we also say as the achromatopsia. So here in normal vision, we can see that all the different cones are normally okay over here, fine over here, but here we can see that all these three cones are lacking. That is, person sees the world only in the shades of gray, black, and white, no color, because only rods or only one type of cone is functional. This is often associated with the poor visual acuity and sensitivity to light. So if we talk about the normal vision, person can differentiate all the colors because the cones and rods they are functional in nature, but when person is just dependent to rod, it is achromatopsia. In prot anomaly you can see there is mild shift in red color, but your it's a lack of alcones and so on. You can see the am cones and as cones they have the mile shift and total lacking over this figure. So this figure is really important. You can take the screenshot of this figure so you can understand color blindness in well manner. Thank you so much for watching this video. You can go through these references if you want to know more about the color vision.